Great pleasure to introduce the last speaker of this session. So Lea Starita has probably traveled the furthest to come here, I think, come all the way from University of Washington over in Seattle. So thank you so much. I must also say, Lea's also been very adventurous. So she turned up in Exeter, got straight on a bus out to Oakhampton and went hiking in Dartmoor. I didn't have anywhere booked and ended up staying in a youth hostel. So anyway, very adventurous. So Lea is a real leader in this field and is going to talk to us about some of the pioneering work she's done on developing tools and techniques for massively parallel functional analyses and effects of variants on protein. So over to you, Lea.
Thank you for that introduction. Can you hear me? Do I have this pinned right? Great. So today I'm going to talk about trying to get to an atlas of variant effects to empower genomic medicine. And the first thing I want to say is I'm going to say we a lot, and everything I'm going to tell you about is in close collaboration with my colleagues Doug Fowler and our clinical partner Andrew Sragatis. Together we've built the variant effects program at the University of Washington. We've assembled a team of researchers, clinicians, and software developers who are behind all of the work I'm going to tell you about today. And I think to start, we can all agree with this statement that using a person's genetic variants to guide medical treatment is a central tenet of genomic medicine, right? We can read genomes cheaply and accurately, and I'm going to talk about the known parts of the genome, not where Wendy goes, but this is protein coding genes. And we can also, I think, agree that precise genetic diagnoses can lead to positive outcomes, right? So from life-saving intervention, this is a Kaplan-Meier curve of people who had risk-reducing surgery versus those who did not, if they carried BRCA1 and 2 variants, pathogenic variants, right? It's a spectrum, and even ending a diagnostic odyssey is a huge relief for many people whose children especially carry or have rare diseases, right? This is actually a fantastic article. It's on the front page of the Washington Post. These are families who have just been diagnosed with the Renew syndrome, which Nikki Whiffen discovered about a year and a half ago now, right? And these families were able to find each other and kind of act as a support group, and for the people who had younger kids, see how the older kids were progressing. So it was just before I started working with these families, like what does it mean that we tell them they have a disease, but we can't do anything about it. It turns out that it's actually a really big deal, and a diagnostic odyssey, at least in the United States, can last for 6 or 7 years, right? So, and also importantly for medicine, understanding variant effects is also the first step toward treatment. When we find a genetic variant in a genome, we have to determine whether or not the variant is going to be pathogenic, which means it's definitively linked to disease, or benign, meaning it's a harmless passenger. But unfortunately, most of the variants wind up in this category here, which is uncertain significance. And these are called variants of uncertain significance. Doctors are not supposed to make any medical decisions based on these, and they're very frustrating to patients who receive them, right? It's very confusing. It turns out that when you tell a person they have a variant of uncertain significance, they hear all sorts of things because they don't understand. And also for the doctors, right? They want to provide optimal care for their patients, and they can't with a variant of uncertain significance. And this is an acute problem because for single nucleotide variants that change amino acids, so missense variants, this is the current accounting of ClinVar, which is the clinical genetics database for Mendelian genes, or variants that may cause Mendelian genes. And if you look at all of the unique variants, about 90% of them are not in definitive pathogenic or benign category. Most of them are variants of uncertain significance, and a decent number of them also have conflicting reports, right? And what's happening also, as sequencing has gotten cheaper, it's being integrated into more and more clinical tests, and this number is rising exponentially, right? So these are unique variants that are found in the human genome. 2025 was not the year that we fixed this problem. And you've got a little bit of a rise in benign variants. This is probably from big population studies like NOMAD. And this also represents, even though they're unique variants, it represents families, it represents people who are not receiving a precise genetic diagnosis. And so how do we find evidence for variant interpretation, right? The old-fashioned way would be segregation. You find these big families, you see if the variant separates, or the variant follows the disease throughout generations. I will tell you, in the American healthcare system, that is not going to happen. And population data, right, where you collect variants from large populations. If variants occur too frequently or more frequently than the disease itself, they can't be disease-causing, they have to be benign, right? Except these two sources of evidence are highly biased by who has had access to genetic testing and who has been included in the population data, right? So we're basically biased by sequencing white people. And then, but on the right side of this, slide are two are variant effect predictors and experimental evidence. And both of these things are scalable. Variant effect predictions can be made for any base change or any amino acid change in the genome. And I'm going to try to convince you today that experimental evidence is also a scalable solution. And by scalable, we are going to use multiplex assays for variant effects. So these are assays where we try to make all possible variants of a gene of interest, or it doesn't have to be a gene, could be an enhancer or promoter. We get those into a cell population, perform some sort of functional assay, and I leave this as a black box because every protein is a special flower. You could also be looking at effects of transcriptional output on a promoter or something like that. But the key to this is that the assay, the genotype is linked to the phenotype such that you can use high throughput sequencing as the readout, right? So I both identify and count variants. And then we calculate scores for the variants, for all of the variants in the assay simultaneously. Most of the time it's some derivation of a frequency of the variant after the selection versus before. And then we map those to the sequence, right? So every nucleotide change will have some outcome. And these are what we call variant sequence function maps, and I'll show you more of those today. So first, I'm going to give you an example of one of these assays and how it can be used for variant interpretation. First, I'm going to tell you about saturation genome editing. So we have this haploid cell line, which is really nice because if you're going to edit, you've only got one target. And it has about 2,000, 2,500 genes that are essential in this line. And so when you edit in a loss of function variant, some of those cells become depleted from the population, and this is something we can track by sequencing. And for saturation genome editing to work, the first gene we really did this on was BRCA1. This was Greg Finley back in the day. And BRCA1 has a couple of domains that are essential. And the first thing we do is we target one of those exons, an exon here, exon 18, with Cas9 and a guide RNA. This causes a double-strand break. We give these cells a library of repair templates on plasmids. So this has basically this sequence plus a single nucleotide variant somewhere. And since we're editing, we can also put them outside of the coding regions in the splice, in the intron-exon boundaries where we find splice variants. And then as the cells use these repair templates to fix this double-strand break, these single nucleotide variants become, integrated into the genome. And each cell then, because it's a haploid genome, has one copy now of a single nucleotide variant in the genome that we engineered. And then we just wait, right? Because these BRCA1, for example, is essential for this cell to grow. And so we see which of these variants become depleted over time. And we sequence the targeted region. We sequence the day 5 cells, and then to see which variants got into the genome. We sequence again at day 11 to see which variants became depleted. And then we generate scores. And for about every 150 bases that we target, we need to do another experiment. So this is where all of the work is. So each of these targets needs a new guide and a new variant variant library, a repair template library. And so we tried to make about 4,000 possible single nucleotide variants, and the scores were pretty bimodal. Here, around log fold change of 0, we had, this is no change, and then becoming depleted over time. And the variants that, the data made sense were synonymous variants that don't change the amino acid, were generally had no change and no depletion, whereas variants like nonsense and canonical splice variants became depleted. But the ones that are hard to predict, these missense variants, were bimodally distributed, where many overlap the synonymous variation, suggesting that they were functionally normal, whereas those many overlap the nonsense variants, suggesting that they were functionally abnormal and loss of function. And importantly, I told you we use this crazy haploid cell line. It used to be a leukemia of some sort. What does that have to do with breast cancer, right? Are we measuring the right thing? And so we mapped that to variants that were BRCA1 variants that were found in ClinVar that were known to be likely pathogenic or likely pathogenic or benign and likely benign. And you can see that this assay separated those really nicely, suggesting that whatever we're measuring as loss of function in the cell line is what's going on in humans whether or not they're at risk for cancer. So this was really exciting to us. And I told you that we wanted to use this data to reclassify variants of uncertain significance. So how did this data perform? Well, it does pretty well. So we use a Bayesian framework. So ClinGen, which is a NHGRI consortium, which kind of makes the rules for how you can translate this data into evidence for clinical variant interpretation. So following their guidelines, we used a Bayesian framework that quantifies how well an assay is able to separate the known benign variants from the known pathogenic variants. And so the more the assay gets right, and the more variants there are, right? So BRCA1 is luckily a well-known or well-studied gene, so there were a lot of controls. And the more the assay gets right, the stronger the evidence can be when you apply it to clinical variant interpretation. And the word strong actually means something in clinical variant interpretation. It carries a lot of weight. Okay, so then we had to collaborate with Ambry Genetics to get their data. So all of the other data that you need for variant interpretation, unfortunately, is not publicly available, generally. So we collaborated with Ambry Genetics, which is a large testing company in the United States, genetic testing company, and they gave us all of their data for which we had a functional score. Their data was about a third of variants of uncertain significance. We applied the functional evidence as strong evidence for toward pathogenicity if there was a loss of function variant, strong evidence for benign if it was a functionally normal variant, and we just used the American College of Medical Genetics Interpretation Framework. I won't get into it, but we were able to reduce the number of variants of uncertain significance by 50%, which was pretty awesome. Most of them went to benign, which is what you expect, but a few went to likely pathogenic, which means that these people are now eligible for risk-reducing surgery, right? And this was a watershed moment for us because we had gone from, fiddling around on the bench, does this work, to Ambry Genetics re-releasing updated reports to all of the doctors who had ordered these tests for these people who had gotten a BUS result, right? So these people now either got updated results saying that they had a benign variant or a pathogenic variant and could then, move on, which was nice. And then recently we've kind of curated data out of the literature for other studies that have done similar things where they have performed these assays on their genes of interest, cancer genes here, immune, all sorts of things, and performed variant interpretation. And kind of across the board, it looks like about 55% of this data is driving greater than 50% variant reinterpretations, which is really great. Another thing we wanted to look at was if we can try to fix this this kind of bias toward European-like genetic ancestry with this data, because as I said, we try to make and measure every possible variant, right? We are not limited by which variants occur in the population. So this study we did through the Atlas of Variant Effects Alliance, and we looked at the variants of uncertain significance in all of us. And for the 18,000th time in genome medicine, we found that the burden of variants of uncertain significance was higher in people who had non-European-like genetic ancestry, right? This was not surprising. But what we did was we then applied the multiplexed data, so data from for 3 genes, BRCA1, P53, and PTEN, so these are all cancer-related. And you can see that before we applied that data before we added that functional data to the clinical variant interpretation framework, we had this bias where, again, there were more variants of uncertain significance in the non-European-like population. When we added the MAVE data, first of all, you can see this huge reduction in the number of VUS, but this bias went away, which was really exciting to us that this technology had gone from the bench, right, so messing around in the lab, to reinterpreted results, and it could help equalize a long-standing disparity in genetic medicine. So we're really excited about this. So we have this tool, and we want to get here, right? We want to get to no more variants of uncertain significance, and how do we get there? And so since these tools are working, scaling up would be one place to go, and we'll talk about our efforts to do that. And so my lab and Doug's lab were part of an NHGRI consortium called the Impact of Genetic Variation on Function, and they have lots of goals. This is basically a follow-on of ENCODE. So ENCODE kind of was a study designed to find all the parts in the genome, and now we're going to break them and see what happens, right? We've got more high-minded goals, but this is basically it. And so my lab and Doug's lab, we kind of launched with 2 production scale MAVEs. One is the saturation genome editing I showed you earlier, and the other is an assay that measures if proteins are unstable. So many pathogenic variants cause proteins to unfold, and that is their pathogenic mechanism. So we have assays that can measure these 2 things. We choose genes based on for multiple clinical uses. First, we have a lot of genes that we've tested that I label with ClinGen, and these are genes that are on genetic testing panels, generally, right? So there's a lot of testing going on, a lot of VUS. The other is for variant discovery for rare disease, and I work with a rare disease consortium called Gregor, where we try to understand some of these variants. Some of these are in diseases that are a little bit more common. Some of these and some of the genes that we're looking at are actually, we're trying to provide evidence to help them with a new gene discovery. And again, it was the same in service of the rare as one population for the Chan Zuckerberg. We use little baby automation in comparison to Nicola, where we're trying to parallelize a lot of the cloning that goes on. Unfortunately, we do large-scale tissue culture. Each one of these experiments is many 10-centimeter, 15-centimeter dishes. But the University of Washington is an undergraduate granting institution. We've got, this is Audrey and Ivan, they're awesome. And I was horrified to learn that when I said this in a different conference, they thought I wasn't paying them. They get paid a lot of money, don't worry. Yeah, Seattle minimum wage is $20 an hour. Okay. So in the last year or 2, my lab has generated SGE scores, saturation genome editing scores for 30,000 variants. Again, many of these are in breast and ovarian cancer genes. This one, that one, these 2. And then these others that are more rare disease. These scores can be highly accurate. I'm just showing you an example of BARD1. BARD1 is BRCA1's best friend. And if we look at the benign variants, they all fall kind of in this functionally normal range, 99.5% of them, whereas 95% of the pathogenic and likely pathogenics fall on the right side of this functionally abnormal line, suggesting that this data is high quality, which is good. And so my lab and Doug's lab together generated 60,000 new variant effect measurements using these two technologies. We also, because the data that has been published by a lot of the field has not been used to its greatest effect. So we curated all of the data out of the literature as well, which adds another 200 or so thousand variants. And then this data set as a whole, totally, covers about 30,000 mapped nomad variants, 25,000 VUS, and 21,000 controls to use. And so what we wanted to ask was, can we leverage this experimental evidence plus variant effect predictors to decrease the number of variants of uncertain significance? And it's kind of controversial in the field. Can you just use these two pieces of evidence? And then, we use them according to the rules that ClinGen has deemed appropriate. And it turns out that just these two pieces of evidence can move 65% of the variants of uncertain significance. Again, most of them go into likely benign. It's easier to get to likely benign, and it should be that way. And we also expect that more variants will go to likely benign. But about 3% go to likely pathogenic. And again, these are the ones that are really going to change medical management. So we'll have to see how that goes going forward. And then I will tell you a short story about new technologies. So the current MAVs that I'm talking about really only work in cancer cell lines, right? We need to change that because there are many genes that are not even expressed in these lines, right? If we're talking about, I'm going to tell you in a second about a gene that is only expressed in cardiomyocytes. So how do we access that? So the idea would be to edit in the stem cell state, push the cells to differentiate, and then be able to assess the variants in those more specialized cell states. And of course, this is going to be hard, right? Stem cells are diploid, which is really annoying. Then you have to get to the differentiation and phenotyping, which can be very difficult. Again, this is Sean Fair, whose project this was. And we're going to use as an example here a gene called MYBPC3. It's a myosin binding protein. It is responsible for about 50% of hereditary cardiomyopathy, which causes heart failure. It is part of the sarcomere, so it's only expressed in cardiomyocytes. It kind of forms a bridge between actin and myosin. And the C10 domain, and it's a very big gene. Right at the end of it is a hotspot for pathogenic missense variants. If you have a loss of function variant in this domain, you end up with this protein can't get into the sarcomere, becomes degraded. Okay, so we devised a targeting technology or targeting strategy where we would knock in GFP to one allele, and this destroys a guide site so that the second allele that has the library, which has all possible variants in an exon, can only go into the second allele, and then that has a red fluorescent protein on it. We edit, again, in the pluripotent stem cells and then differentiate to cardiomyocytes. This is what this kind of looks like. So edited iPSCs kind of grow in colonies. We do a cardiomyocyte differentiation, so you can see these cells have gotten big and square. The wild type allele is marked with GFP, and you can see the striated, the sarcomere here. And then some of these variants look just like the wild type, and then some of them look like they're gone, which is to be expected. And so the way that we can quantify this is using fact sorting. So we sort into bins by brightness, and then we sequence which variants are in which bins. So the variant, and we do a weighted average. So the variants with high scores are going to be the bright ones. The variants with low scores are going to be the lowly expressed ones. And first of all, we were super excited because we targeted 2 different exons in 2 different experiments. And we were able to do this for about 1,100 variants, which was really awesome because before this, only about 50 to 100 variants had ever been edited into these stem cells before, so this was great. We were able to separate synonymous variants from nonsense and canonical splice variants. And then again, you can see these loss of function missense variants, which are the ones we were looking for. This assay accurately identifies pathogenic and benign variants. This is actually strong evidence. It's got enough variants on both sides. And then these are 3 cases of Andrew's patients who are now eligible. This is the UW Adult Genetics Clinic. We were able to reclassify them as likely pathogenic, and these people are now eligible for advanced care, which is great. All right. So we have new technologies, and we've got a couple of other genes. Hopefully, that preprint of the stem cell work will be ready to go soon, or I will kill somebody. The next thing we need to do is community building, right? So we are now we helped to co-found the Atlas of Variant Effects Alliance. Jonathan Mill is now on the executive committee. And this is some of our propaganda. You can imagine a future where we're kind of browsing through variant effect maps to understand what any possible change in the genome might be. And we're trying to propel the generation and use of these. So we're trying to bring together data generators, curators, consumers, a lot like this meeting today. We were founded in 2020. We have 700 members already, and I think folks in this community should definitely join us. We host the Mutational Scanning Symposium. The next one will be March 2026 in Melbourne. We host a variant effects seminar series. This is run by trainees for early career researchers, and we also build resources and standards for the use of functional data. And then just real quick, the last mile. So it turns out we figured we were generating all this really useful clinical data. People weren't using it, and we were wondering why. And we found out that if you stuff your data in supplementary table 2, that clinicians can't find it. Okay? And so we're trying to figure that out. And then the other thing we asked clinicians was, how do you feel about multiplex assay data? And it turns out that in Australia, this is led by Rhiannon Villani, and she's here, in the United States, and in the UK, in a study led by Claire Turnbull, it turns out that people who are actually using this data in the clinic are terrified of these assays. Okay, they're complicated. They don't know what it means, and so we've got a lot of work cut out for us here. I told you about kind of how we calibrate these assays. Everybody in the UK just wants Claire Turnbull to do it for them. That's what this data says. Okay, so what we're going to try to do is get this data into ClinVar, and also then link it back to a database where people can, have a dashboard for each gene, each variant, to kind of help them understand what the data means. And so this is about to be launched, and we're always looking for beta testers for this. So in conclusion, we now have about the 1% of an atlas of variant effects. This is just the number of clinical tests, so basically clinical testing volume on the bottom versus the number of SNVs, and we've got data for about 41, 42 clinically relevant genes. There are about 4,000 genes that are definitively linked to genetic disease. So we're getting there, right? So the atlas at 1% now, and hopefully we get to this atlas of the future where if anybody ever needs a variant effect, they can come and look it up in a map in the atlas. So with that, a lot of people to thank, and I can take questions if there's time.
Thank you very much, Lea. That was a really brilliant presentation.